Hello, everyone. Welcome to this webinar. Let me introduce myself first, because this is the first webinar we are doing together. So my name is Galo Goj. I'm a postdoctoral scientist at the Swiss Tropical and Public Health Institute, where I study transmission of tuberculosis and evolution of drug resistance, which are the topics that we will be talking about. So in this first webinar, we will be talking about how to predict phenotypic drug resistance based on whole genome sequencing. As you may know, drug treatment for tuberculosis consists in so first-line treatment for tuberculosis consists in a combination of 4 drugs, of which rifampicin and isoniazid are the main drugs used in the treatment. And thus, if we have this disease presentation or the bacteria that is causing the disease, which are resistant to rifampicin, then we say that it is a case of rifampicin resistant tuberculosis or rifampicin monoresistant tuberculosis. When there are additional resistance to isoniazid, we classify that case as multi-drug resistant tuberculosis. And then we can also talk about extensive drug-resistant tuberculosis if it's a case of multi-drug resistance, plus the bacteria that is causing the disease has additional resistance to fluoroquinolone, to at least one fluoroquinolone, and one of the drugs of the group A, such as bedaquiline. And as you might have noticed, this definition here is the definition of following the new guidelines of the WHO, as previous definitions were based, for example, taking into account second-line injectable drugs such as kanamycin or amikacin, etc. So, and also remember that when we are talking about TB, we can either talk about the disease, so tuberculosis, of the bacteria that causes disease, which is Mycobacterium tuberculosis. Okay, so it can be rifampicin, a case of rifampicin-resistant tuberculosis that would be caused by rifampicin-resistant Mycobacterium tuberculosis bacteria. So when we want to use whole genome sequencing to predict phenotypic drug resistance, what we want to do is basically the following. So we would have susceptible bacteria, and that susceptible bacteria, we want to analyze the genetic content and thus be able to say, well, this bacteria is most likely susceptible. And we will be able to, by analyzing the genetic content of the bacteria, to find something in the genome that tells us that this bacteria is most likely rifampicin resistant or isoniazid resistant or maybe multi-drug resistant. Now, you might have noticed that this schematic representation of Mycobacterium tuberculosis is incorrect, and I did this on purpose, and the reason it's incorrect is because Mycobacterium tuberculosis bacteria do not have plasmids. So Mycobacterium tuberculosis does not present plasmids, does not present horizontal gene transfer as far as I know, and thus this means that evolution also of drug resistance occurs mainly by chromosomal point mutations. And that makes that, as compared to other bacteria, predicting drug resistance in Mycobacterium tuberculosis based on the genotype is much more easy. And the reason is that we have bacteria with just one genetic element, which is the chromosome. And then what we want to do is to be able to detect particular mutations that we know that are associated to different drug-resistant phenotypes, right? So in this case, we would analyze the genome, we will find this mutation that confers resistant to rifampicin, and then we would say, well, this bacteria is most likely rifampicin resistant. This might be another mutation that confers resistant to isoniazid, or maybe the bacteria have both mutations, and then we know that bacteria is multi-drug resistant. And we will see how we know these mutations and how we detect them in just a few seconds. So first, I would like to introduce something, although very basic. I think it's important. So you know that the way drug resistance works is we have discovered different drugs that we use for to treat the disease, and the way these drugs work is by targeting essential functions or essential structures or essential pathways for the bacteria that is causing the disease. So, for example, we know that rifampicin targets the RNA polymerase beta subunit, so precluding the RNA polymerase to work properly. Or isoniazid, it's activated. Well, you know, isoniazid is a prodrug, so it needs to be first activated by the catalase of mycobacterium tuberculosis, and then its active form targets the protein reductase that is involved in the cell wall biosynthesis. And this happens for all drugs. So basically, they target essential functions. And these functions or structures of the cell are encoded by genes, right? And these genes might have some mutations that happen through evolution. Some, and I want to highlight the word some of these mutations, like for example, this is arguably one of the most known mutations conferring rifampicin resistance because it's the most common. So RPOB S450L, for example, is a mutation in the RNA polymerase beta subunit that makes rifampicin to not be able to bind to the beta subunit, and then rifampicin cannot exert its function, its effect in the bacteria. But it is important to know that whereas we have a catalog of certain mutations that we know that are involved in drug resistance, not all mutations in genes that are involved in drug resistance will cause drug resistance. Meaning, we know there are many mutations in RPOB, for example, that cause drug resistance to rifampicin, but that doesn't mean that all mutations in RPOB can cause drug resistance to rifampicin. The reason is that, for example, you might imagine that when one mutation can appear in RPOB, and that mutation might be a synonymous mutation, for example, or even if it's a non-synonymous mutation, by the way the conformational changes that it will produce in the protein, maybe those changes do not impede rifampicin to bind to the beta subunit of the RNA polymerase, and maybe rifampicin can block the activity despite a mutation in RPOB, whereas other mutations, such as this one, produce a change that impedes rifampicin to bind to the beta subunit. So take-home message of these slides is, remember, not all mutations in a gene that might be involved in drug resistance, not all mutations cause drug resistance. And this is important when we are trying to predict drug resistance, because we need to build first, and I will explain now how, we need to build a catalog of mutations that we know they confer drug resistance. How do we do this? So just imagine we collect a bunch of resistant strains. Okay, these are Mycobacterium tuberculosis strains that we know they are resistant because we have performed, phenotypic drug susceptibility testing, for example, with MICs, and we know these bacteria, these strains are resistant to a particular drug, let's say rifampicin. And these strains, all of them carry this mutation. This is just a dummy example with color-coded mutation. So all these strains carry a red mutation. And we know that all these strains are resistant to one drug. So we would say, well, this red mutation is a high confidence resistant mutation because every time I observe strain with this mutation, that strain turns out to be resistant to this particular drug. It might happen in other cases that we have other mutations, like this blue mutation, in which, most strains carrying this mutation are resistant, but some of them are not resistant. Actually, they are susceptible. So then we would say, well, this is, I would say this is a low confidence resistant mutation, because yes, it is true, many of the strains, when they present this mutation, can be resistant, but I also know there are some strains with this mutation that are susceptible. So then I'm not sure anymore about whether this mutation is really involved or directly involved or clearly involved in rendering this strain drug resistant. And of course, we can have other mutations, like this yellow mutation, in which most of the strains are susceptible. And then we would say, well, most likely this mutation is not associated to drug resistance because most of the strains carrying this mutation are susceptible. So we go from phenotypic information to genotypic information. So now we know which mutations are associated to drug resistance and which are not associated to drug resistance. So these would be like our catalog of drug resistance conferring mutations. And what we want to do is then, with such catalogs, when we perform whole genome sequencing, to be able to say, well, I have this bacteria here, I analyzed the genome, I observed it has a red mutation, this red mutation. So then I'm pretty sure this is a resistant bacteria. Or it has this blue mutation. So then, well, it's likely resistant bacteria, but I am not sure anymore. Or it has this yellow mutation. Well, it's likely susceptible bacteria, so maybe we can treat the patient with the regular treatment. Now, how do we build this catalog? So of course you don't need to build these catalogs. So thanks to huge efforts done by several consortia and in general by the community of tuberculosis scientists, now we have this catalog. So we have evidence that comes from laboratory experiments where the particular mechanism of resistance of a mutation has been shown. On the one hand, that's one type of evidence. And the other type of evidence, which are these catalogs that you can see in this slide, is collections of thousands and thousands of strains with their associated phenotypic drug susceptibility data, like MICs, for example, minimum inhibitory concentrations. And then they also have analyzed the genomes of that bacteria. So basically, it's like in this strain, is this effort done here, but just at a huge scale with thousands of bacteria. And then that includes, this is actually very nice, that since 2021, the World Health Organization has released a catalog of such drug-resistant conferring mutations that you can use in your research. You can just go to the webpage and download, click here, the link here, download the catalog. And if you do so, you will see a catalog like this one. So for each drug, here's rifampicin, you will have all possible mutations that have been found or assessed. And then you can find how many strains that carry this mutation are actually phenotypically susceptible, how many are phenotypically resistant. And based on that, you have the sensitivity, the specificity, positive predictive value, and so on. So you can use this catalog. So, you would analyze the genome, you will detect the mutation, will look for these mutations here, and you will say, okay, so this strain is most likely resistant, or this strain is most likely susceptible, which is basically the process of whole genome sequencing, which is I just said. So now what we want to do is we have the bacteria. That bacteria has a chromosome, so it has a genome. And what we want to do is to analyze this genome and find all mutations of this bacteria. And then we collect all the mutations that bacteria have. And now we can compare all the mutations through these catalogs that we have been talking about. And now we can see, okay, so actually this bacteria has this mutation that when I look in the catalog confers isoniazid resistance, and also has this mutation that when I look in the catalog confers rifampicin resistance. So I'm pretty sure this bacteria is an MDR strain, and thus it will be resistant to both isoniazid, rifampicin, and I can, depending on what I'm doing, either research or taking clinical decisions, I now have this valuable information based on whole genome sequencing. And this is pretty much what you will be doing in the hands-on tutorials. So basically you will be analyzing bacteria, you will be taking the catalog of mutations that you will find in all the different bacteria, and then you will be using TB profiler to compare the mutations to a catalog, and then to say all these strains are susceptible, these ones are rifampicin resistant, these ones are multi-drug resistant. So that was all for this webinar. In the next webinar, we will be shortly talking about phylogenetic mutations. It's not something we will be talking really in depth, but we believe it's important that you know that they exist and you know what they mean, because they might be important for your research, or you might find them when reading papers, and you need to know what they mean in the context of drug resistance. So that was all. Thank you very much, and see you in the next webinar.